Have you ever wondered how cells are able to reproduce? Well, there are two different ways cells replicate and reproduce. They're mitosis and meiosis. However, mitosis and meiosis have several differences that contribute toward duplicating cells. The most prominent difference between mitosis and meiosis is that meiosis occurs in both haploid and diploid cells, whilst meiosis occurs in only diploid cells. Haploid cells are one pair of chromosomes, while diploid cells are two pairs of chromosomes. The mitosis cycle results in the creation of two daughter cells, in which each is genetically identical to the parent cell with the same number of chromosomes. The meiosis cycle results in the creation of four daughter cells, in which each haploid are genetically different from each other and from the parent cell.
Meiosis is part of the process of sexual reproduction. Sexual reproduction begins with the process of fertilization that results in a diploid zygote for multicellular organisms because it has two haploid sets of chromosomes that have genes of the maternal and paternal family lines. The zygotes then go through mitosis and with the help of descendant cells generate the somatic cells of the body. Somatic cells are any cells in a multicellular organism other than gametes or their precursors. After mitosis, gametes, which are reproductive cells that transmit genes from one generation to the next, are produced through meiosis. Fertilization and meiosis alternate in sexual life cycles and help maintain a constant number of chromosomes in a species from one generation to the next.
Let's begin by explaining the stages of meiosis. Meiosis has two cycles of cell duplication, meiosis 1 and meiosis 2. Before meiosis, cell cycle occurs in interphase. During interphase, DNA replication occurs with uncondensed chromatins. Also, the centrioles and aster form within the cell, and nucleolus is present within the cell. During this phase, the cell also obtains nutrients and proteins in order to grow and conduct normal cell functions. Most of the cells spend their lives in interphase to grow, and then during mitosis and meiosis undergo division. After interphase, the cell goes into prophase 1. In prophase 1, the nuclear envelope breaks down as the chromatin condenses. The mitotic spindle begins to form, as shown in this clip. The sister chromatids are attached to proteins called cohesins. In the early stages of prophase, a homolog aligns with the corresponding genes on another homolog. Homologous chromosomes, or otherwise homologs, are paired chromosomes that have the same length and centromere position that carry genes controlling the same inherited traits. Each chromosome pairs with a homolog while crossing over occurs and initiates meta. Crossing over is the exchange of genetic material between non-sister chromatids. After crossing over, the homologous pair moves toward the metaphase 1 plate. The movement between the metaphase plate acts as a substep between prophase and metaphase, called prometaphase 1. During prometaphase 1, the nuclear envelope completely breaks down, and a pair of sister chromatids connected by kinetophores around the chromomere. Kinetophores are the protein structures on chromatids where spindle fibers attach. The kinetophore microtubules extend from the sides of the opposite poles and attach to kinetophores. After prometaphase, the two homologs line up at the metaphase plate in metaphase 1. Microtubules are attached to kinetophores at the centromere of both chromatids within one homologous chromosome. In this cell, 4 homologous chromosomes are lined up along the metaphase plate. The next step that the cell goes through after metaphase is the process of anaphase. In anaphase 1, the homologous chromosomes separate and move towards opposite poles guided by the mitotic spindle. The sister chromatids remain attached throughout the entire process, so the whole chromatids move as one unit towards the poles. Anaphase sets up the action for the last stage of the meiosis. Telophase is the last phase in which a nuclear membrane begins to form once again, and the two cells split to create two haploid daughter cells. Cytokinesis, which is not part of meiosis, is another event that occurs simultaneously with telophase 1. New chromosome duplication occurs between meiosis 1 and meiosis 2.
Now the cell goes through prophase 2, in which the spindle apparatus forms, and chromosomes condense to go into prometaphase 2. Similar to prometaphase 1, two chromatids at the centromere move towards the metaphase 2 plate. The metaphase 2 plate isn't an actual plate, as we said before, but it is a line of representation. Also, in prometaphase 2, a pair of sister chromatids is attached to both poles. Now the cell enters metaphase 2. The chromosomes line up at the metaphase plate. The kinetochores of sister chromatids attach to microtubules that extend from opposite poles. The two sister chromatids of each chromosome are not genetically identical this time because of the crossing over that occurred in meiosis I. After metaphase 2, the cell engages in anaphase 2. The breakdown of proteins holding the sister chromatids at the centromere allows the chromatids to separate and the chromatids move towards opposite poles as individual chromosomes. After anaphase 2, the cell reaches telophase 2, where the nuclei form and chromosomes begin decondensing. Four daughter cells with a haploid set of chromosomes are produced at the end of this phase. The four daughter cells are genetically distinct from one another and from the parent cell.